Hello and welcome to Greiner Bio-One Talks. My name is Ana Maria Simundic, and I am a Global Director of the Department for Medical and Clinical Affairs at Greiner Bio-One. And we are here today to talk about hemolysis. And I have a special guest with me, and that is Dr. Kendall Cradic. Welcome, Dr. Cradic. Dr. Cradic works at the CORPath Pathology Services in Columbus, Ohio. And besides being a fantastic scientist and a professional, in his free time, he enjoys woodworking and tackling renovation projects. And he lives in Columbus, Ohio with his wife and 3 sons. So welcome once again, Dr. Cradic.
Thank you.
And tell us a bit about where and how and why your journey began at Mayo Clinic.
Yeah. And first of all, thank you for having me. I'm happy to have this discussion with you. I got my start at Mayo Clinic a few years ago. I was very lucky to land there doing research and development, and we were developing clinical assays. I started in molecular work, so working with sequencing assays, reverse transcription, PCR, and some quantitative PCR. And then I also got to work with a team where we developed immunoassays as well as mass spec assays. So I had a pretty wide scope of experience there, and I was just lucky to be part of that team and to just get to do a lot of very fun things.
That is so exciting. And tell us a bit about your current role at the CORPath Pathology Services.
Yeah. So currently, I am serving as a medical director for a medium to large size system in Ohio. We have about 16 hospitals and a number of freestanding emergency departments that we provide laboratory services for. And so I oversee chemistry, and I get pulled into a lot of other areas like point of care and some of the other areas that are closely related to chemistry. But there's a lot of core labs, a lot of different types of collection and testing, and all of the analytic workflow processes that are going on in that system. So, yeah.
So I think you're the right person to talk about hemolysis, which is the most, and probably the most frequent and the most challenging preanalytical issue. So tell us, maybe let's start with the definition of hemolysis. So how do we define hemolysis, what it is, and how do we detect it today? So what are the requirements from the regulatory standpoint? How do we need to manage hemolyzed samples in terms of the detection?
Mm-hmm. Yeah, requirements from CAP state that we need to assess our specimens for quality. And one of the main drivers of poor quality is hemolyzed samples, where the red blood cells are rupturing for any number of reasons. This can happen, and they leak their contents out into the serum or plasma. And the contents, the electrolytes, the proteins can interfere with other assays and can cause false elevations or false decreases in those other results. And so we have to be very careful that we are assessing samples that are hemolyzed and that we are either not reporting results downstream, or if we need to report a result, we are doing it with appropriate safeguards in place.
So what are the challenges in detecting hemolyzed samples? Or maybe let's first start with, how do you detect hemolysis in your lab? And then what challenges do you see in that approach?
Yeah. So our lab is highly automated. So most of the core labs that we have automated detection of hemolysis. So almost every specimen that's going through is getting an assessment by the instrument of what the level of hemolysis is in that sample. But that's not true across the board. There are a lot of labs that don't have fully automated. They may have semi-automated processes or semi-quantitative processes for assessing hemolysis. And even some labs are limited to like a visual assessment of the sample and maybe comparing a hemolyzed sample to a color chart. And so the challenges are getting all of these methods standardized, for one. It's hard to get a standard assessment of hemolysis when you're having a visual comparison compared to an actual assay for color detection or hemoglobin. So it's that's one challenge there. You can't just have a single assessment of hemolysis from one lab or one method to another. Another challenge that I think is out there is just the sheer number of specimens that we deal with, and every one of them is being assessed for hemolysis. It's like adding another test on top of the whole entire menu that we have, and but that's how important it is. So we go ahead and do that.
So may I maybe go back to that standardization, because I think it's really important. So yes, you mentioned that there is a big difference between visual and automated detection, but we know that also there is a lot of variation between different manufacturers offering automated solutions.
Yes.
And then it's not only the different methodology, but also different cutoffs. And so could you comment on that? How do you see, what is the way forward? How do we address that?
Yeah, and that's particularly troublesome in systems like my system is not completely standardized on one analytical platform. So in some labs we have automated platforms that are using different measuring units than other labs, and so we can't have a consistent assessment of that hemolysis. So the way we would warn or the way we would assess that sample and the effects in one lab might be different than the other, and it's very challenging because there is no standardization between those two. I think that that's one area where we could probably see some improvement in quality across all labs is if there was some kind of a standard assessment for hemolysis, so that no matter what assay was being performed, we were at least having the same assessment of what that sample quality is, no matter irregardless of the platform.
Exactly. And then again, there is also a lot of heterogeneity in acceptance criteria or decision limits about what is acceptable for which parameter. And yes, manufacturers do provide their own values, but do you think that these should be verified in the lab? And do you verify? Did you verify those in your lab?
Yeah, that's a good question. And the answer to that is sometimes. We in my lab, we have done verifications of those limits, but it's true. Each assay is going to be, it's going to be affected independently from other assays by the amount of hemolysis. And so where we have critical assays that are very affected by hemolysis, I've gone ahead and had my staff do the interference studies so that I can understand exactly what's happening with those samples, not only the trend, but also the variability, because not every serum or plasma sample is the same. Sometimes a little bit of hemolysis will affect an assay a great deal, but then the same assay with a different hemolysis and a different plasma may affect that result differently or to a lesser degree. So it's not just the trend, but also the range of interference that's important. So when we're trying to advise physicians downstream, we give them a result, and if that sample was hemolyzed, we may have to say this result may be falsely elevated or falsely decreased by such and such a percentage. So it can be very tricky, especially like you say in without that standardized piece.
So what is, in your opinion, the role of manufacturers in that respect? Because labs actually can only rely on the manufacturer's declarations, and sometimes they're not so suitable to be used in everyday work. So then laboratories are actually required to put a lot of effort and time and money to do their own verification studies, as you already said. So what is, in your opinion, the role of manufacturers? How can manufacturers can help to address that issue?
Yeah, I think it would, if the manufacturers could do more, that would, I think, increase the quality of the assays that they're selling, for one, and also the quality of the results that we're producing for the patients. I don't know what the motivation for manufacturers to do more is. I don't know if there's any any regulations or potential regulations that would force them to do more. I think it would have to be more or less voluntary at this point. But I do know that the companies that are producing more helpful assessment of hemolysis are definitely, I think, leading the curve in providing us with good tools for giving quality patient results. I think it is important. I think that I don't want to call out any one manufacturer or another. I think they're all doing what they can, and I think that they all need to do much more, similar to the way they give us reference intervals to the best of their ability. I think that this is like kind of the next step, that they could give us more information about interferences that they know or that they have found that affect their assays.
Thank you. So let's move then to hemolysis prevention. So could you let's first start with major causes of hemolysis. What are the major causes, and then how do we prevent hemolysis?
Yeah, I think in my experience, I've seen two areas that are probably the most problematic for causing hemolysis, and that is collection technique, or the people that are collecting through IVs or using really narrow gauge needles when they probably should be using a wider gauge needle, and collecting into syringes and things like that. So those collection techniques is one thing. The second is transport. The samples, when they're being sent through pneumatic tubes or sitting around in pickup boxes and then shipped off via couriers in the back of a truck or who knows where, all of these things cause red cell lysis, and then you end up with hemolysis. So if the sample's not adequately processed before transport, or if the collection is poor, then these are, I think, the two main drivers of hemolysis that I see in my labs.
So how did you address these challenges in your lab?
Yeah, so we tried to address each of those separately. So the first piece, and the one that I think is most exciting, is I've been working on a bit of a program. It's educational and also metric-based program to help our collectors get better at collecting. So that technique-oriented piece. So we've developed a scorecard that we use, that we assess hemolysis and other areas of sample quality that we can measure. And we review that scorecard with every single collector as they're collecting throughout each month. So at the end of the month, they can see how they did in relation to everyone at their site, and then in relation to everyone globally in the system. And it's been used very effectively. We've seen hemolysis rates drop. We've seen the quality of specimens go up. And we don't use it in a punitive way at all. It's a very educational tool, and it's really, really helped just reduce our redraw rates and are, reordering, recollecting. So it's been a great success so far.
That is exciting, really. It's like the use of indicators. I mean, in other areas, it's about benchmarking yourself. And what is your hemolysis score?
Yeah,
Right?
exactly.
Yeah.
And it's what's really interesting to me is how much information you can learn just by counting things.
Yes, exactly.
Counting.
Yes. If you don't, you cannot improve what you don't count, what you
That's
don't
right.
measure, right?
That's right.
Exactly.
Yep.
Okay, well, let's. You did touch upon a bit about on results reporting. So how do
Yes.
how should we report hemolyzed results? So let's start with recommendations and guidelines standards. What do they say about reporting results from unsuitable specimens?
Yeah, I think I don't know the exact wording of the regulations, but I think that the main thrust of this is that we are supposed to be aware and very proactive about making sure that the results we report are not influenced adversely by any interfering substance, as to the best of our ability. And hemolysis is one of those. And so there are some specific checklist items I know about managing the quality and monitoring that as we produce results. And hemolysis certainly fits in that category. So we do that, and we take it very seriously. We are constantly getting better at this. It's an ongoing struggle to understand how to report results that are hemolyzed, when you can do that safely, and when the sample should just be rejected outright. So for our main sort of set of just garden variety tests, we have limits that are either set by the manufacturer and verified, or we go beyond and do additional studies ourselves sometimes so that we understand where those limits should be set in the population that they're being used. So our techs can be empowered to either release the result with a comment that indicates what direction and what scope that result may be influenced by, or to reject the sample and call for a redraw. So that's kind of the basic strategy that we use, is that we have those thresholds. We report results with comments sometimes, or we reject the sample if the sample is too hemolyzed. And then we have another category altogether, and that is the sort of the irretrievable specimen, or the something that is too precious to just throw away. And so we might take like a fluid collection, for instance, and it may be hemolyzed, in which case the result may still be useful as long as the physician knows the probable influence that hemolysis has on the fluid. So we try to report that appropriately. And sometimes that just means picking up the phone and having a conversation with the physician before we report the result.
So rejecting a hemolyzed sample is never an easy decision, right? Because you always have in mind how difficult was it to collect it. So try to think, like, how is it going to be 10 years from now when, there are some new solutions? How do you see this problem solved? And there are many areas where this problem might be solved. So what is your, idea of?
Yeah, I think that's a tough question. I think there are a couple of answers to that. I think one is that I've thought about ways that we could, for the assays that are strictly influenced by either the color of the hemoglobin, so spectrophotometrically, or I think I've thought about ways we could try to remove the hemoglobin from the sample, and maybe that will become a reality at some point. I know that's very challenging right now, strictly because there's just so much hemoglobin in there, and it's hard to selectively remove that.
That's an exciting idea.
Yeah, I've thought about that quite a bit. But other ways, there are other ways that assays may be able to improve. I know one thing that has been very useful is that the assays that we're running now compared to 20 years ago are using much less sample, and the dilution factor is larger than it used to be. And that in itself helps because the assay has gotten so sensitive that we can do that dilution and get rid of interfering substances or get them reduced to where they're not interfering anymore, and then get a quality result, even though that interference was present at the physiologic concentration. So those are a couple of areas that I've thought about that I think there's probably still room to go. Yeah.
Okay. Thank you. Well, this has been very exciting. Thank you for sharing with us your thoughts and ideas about hemolysis. So maybe just one last question before we end this podcast. You obviously have a long experience in the lab. So could you share with us maybe some memorable pre-analytical experience that had something to do with patient and from a patient perspective?
Yeah, the story that comes to mind it was kind of humorous for me because all my training and where I worked, we always had electronic orders and everything was electronic. And when I moved to a new system, we had my wife had to go to a specialist, and she came home with a paper requisition for a lab test, and I was.
You probably haven't seen it ever.
I didn't know what to do with it. I had to actually call one of my managers and ask her, how do I get this paper requisition turned into an actual blood collection? So I look back at that and I laugh, and I think sometimes, things aren't, the rest of the world is not always the way you think it is. There are different pockets of workflows going on out there, and that's okay. But I'm always learning something new, and I thought that was a interesting and humorous experience.
So thank you once again for a very interesting discussion about hemolysis. So this was episode on hemolysis, and you were listening to Greiner Bio-One Talks.